a inhibitor. So what are the differences between a drug and inhibitor? So it should be highly selective and it should be less toxic. Okay, and it should be bioavailable. Bioavailable means, for example, you might have seen on a drugs when you take a paracetamol, 250 mg, 500 mg, they call it as a mg. That is in a milligrams. That particular active pharmaceutical ingredient API, it is called as API, which is present in a paracetamol drug, along with the other excipients, other APIs. So that particular drug is present in a that quantity, 250 mg to 500 mg. So bioavailability means when you take a 250 mg tablet, it should be maximum available to the inside the in vivo system to show the efficacy. If you take 100 mg and if only 20 mg is reaching the particular active site and remaining 80 mg is not reaching the particular active site due to some side effects, it is not a good bioavailable drug. So it should be, the drug should be a very good bioavailability and it should reach the target site. For example, what is a target site? So when we have a hand pain or a knee pain, for example, sportsman, we have seen in the recent, I think many of you have seen in the cricket last test match, many of the players are got injured because they may have some ankle pain or a wrist pain, something like that. But what we do, we take some spray or we take some medicines, oral tablets. But it reaches to the hand or it reaches to the leg or it reaches to the some other the particular active site where the pain is. So that is called as it should reach to the active site. Even though you are taking a tablet orally, but it knows the tablet has been or the drug has been designed in such a way it can reach to the active site. It can identify where is the pain, where is the inflammation, where is the particular problem. So in that way, the drug should reach an active target. Then ease of synthesis. If you have a very good activity, so if you have a very good activity and if the synthesis is taking a long steps and it is a 50 steps and if you want to spend a lot of time, so there is no use. So it should be easily synthesizable. It is cost effective. It should be very low price because, for example, country like us, we are a developing country. We have a more lower income people than compared to the other countries. So country like us, we need a very less or very low cost effective processes, cost effective drugs, affordable drugs where a common people can afford. So that is the one of the parameter. And stability of on storage. It should be stable when you store it at a room temperature. Okay. Then pharmacokinetics of a parameters, then no allergies or side effects. These are the properties which differ from basically from the drug to inhibitor. Then when a molecule is discovered in a lab, either it is in a university or a national institute, anywhere from the world, when you prepare this molecule or discover this particular molecule in the first stage, what we see is whether it is obeys all the ADMET properties. The ADMET properties called as a pharmacokinetic properties. Those properties are called absorption. Absorption means when you take a orally, in particularly ADMET properties obey with the orally available drugs. When you take the tablet or the syrup in the child in the children, we give usually syrups. So when you take the particular medicine, it should be absorbed properly in our glut. Then from there it should have a distribution pattern equally at a particular active site. It should travel and it should have both lipophilicity character and lipophobic character, so that it will travel easily to the active site. And after that, reaching to the particular active site, it shows the activity. Then after that, after some time, it should start metabolism. The drug, it should not stay longer time in the active site or in a human body. It should metabolize. It should metabolize through the enzymatic process. The enzymes, I will just give you in the coming slides what are the enzymes, how the drugs are converted into, or how the drugs are metabolized. In chemistry lab, we can actually convert some molecules into another molecules, oxidation, reduction and all. But what is happening inside